Hello my dear students, this is Indira, your botany lecturer. Welcome back to my channel. Those who are watching for the first time, please do subscribe my channel so that you will be getting all my updates. And now coming back to the topic, in today's session, I am going to discuss about the packaging of DNA helix. Meaning of what is packaging of DNA helix is DNA is a very long molecule. We already discussed the length of DNA is measured in terms of base pairs or in terms of nanometers. And this long DNA or DNA should be packed into a very small cell, and within a small cell it has to be packed into a very small nucleus. In human cell or in general, the size of eukaryotic nucleus is about 10 to the power of minus 6 nanometers. Meters. This is the size of average size of nucleus. So in this size of nucleus, the DNA has to be packed so that it can be visible as compact chromosomes in the nucleus. In nucleus we find chromosomes which are in the form of chromatin, and this compact chromatin in the nucleus is because of the DNA packing. So now let us see what is the packing and why this packaging is required. So if we want to measure the length of any DNA, length of DNA can be measured by multiplying two factors, that is distance between the two base pairs or nucleotide, distance between two base pairs or nucleotide pairs, which is about 0.03 or 0.34 into 10 to the power of minus 9 meters. What is this distance? It is the distance between two base pairs of a strand, two base pairs. This should be multiplied by total number of base pairs present, This is how we can calculate the length of any DNA. So taking this concept, let us calculate the length for human diploid cell. In a human diploid cell, we find the number of base pairs are 6.6 into 10 to the power of 9 base pairs. This is per diploid cell. So this is the total base pairs that should be multiplied by this length, that is multiplied by 0.34 into 10 to the power of minus 9 meters. So if we multiply, this will give approximately about 2.2 meters. This will give approximately the length of about 2.2 meters. So this length of 2.2 meters of a cell should be packed into such a small size of the nucleus. That's how the packaging of DNA is required. Now if we calculate similarly for another example, that is for E. coli. So in E. coli cell, we have number of base pairs is equal to 4.6 into 10 to the power of 6 base pairs. Again, this should be multiplied by this distance, 0.34 into 10 to the power of minus 9 meters. This will give approximately a length of about 1.36 millimeters. So in this way, if we know the number of base pairs for any cell, for any organism, we can calculate its length of the DNA. So to accommodate such a long DNA into a small nucleus, the packaging is required. This DNA packaging is observed both in prokaryotes as well as in eukaryotes. Eukaryotes, definitely it is more complex. It has because a complex genome. So eukaryotic packaging is more complex compared to prokaryotes. So now let us see first what is a DNA packaging in prokaryotes. Let us see first DNA packaging in prokaryotes. So how it happens in case of prokaryotes? So in prokaryotes we know there is a nucleoid present. Nucleoid is found in prokaryote. That means this nucleoid consists of a coiled DNA, a circular DNA, or a coiled DNA, circular DNA. And we also know this circular DNA is not scattered throughout cytoplasm. It is present in a place which is called as a nucleoid. So this long DNA will form loops. It forms such a loops and then it remains within a particular place which is called as a nucleoid. So this is called as packaging in prokaryotes. So how this happens? Here DNA is associated with, in case of prokaryotes, DNA is associated with non-histone proteins. Non-histone proteins to form this coiled or supercoiled structure and remains within one place, that is called as a nucleoid. These non-histone proteins are polyamines. These are a type of proteins which are present here called as polyamines. Now these polyamines are associated with DNA to form nucleoid. And RNA also may be involved here. RNA is also involved. RNA is also involved here. This is how the packaging is done in case of prokaryotes. Now let us see the packaging in case of eukaryotes. So let us see packaging in eukaryotes. Eukaryotes, it is more complex way of packaging is seen because of many chromosomes present. Eukaryotic cell will have many chromosomes. So there will be a complex packaging methods are seen. So in eukaryotes, the packaging is explained with the help of two models. Here I am going to explain two models of packaging. In the first model, first I will write down what are those two models. First model is Nucleosome Model. First one is Nucleosome Model. The second one is called as a Solenoid Model. Solenoid Model. So what are these and what is the relation between these two? We are going to discuss. So first let us talk about nucleosome and its structure and all details about nucleosome. Nucleosome. So this nucleosome model was proposed by, first I will give you the scientist names. The nucleosome model it is called. This model was proposed by Kornberg and Thomas. They proposed this model of nucleosome. It is the most accepted model. And according to this model, the nucleosomes are described as beads on string. So in electron microscopic structure, these appear as beads on string and they form chromatin. They appear as beads on string and they form chromatin. So that is how it is described. And these beads which appear as, these structures which appear as bead like, these beads are named as nucleosomes. So if we observe chromatin under electron microscope, it looks like this, a beaded appearance. It looks like a beaded appearance. And each bead represents a nucleosome and a chromatin thread. It consists of a chromatin thread. So this is how beads on string structure is explained for nucleosome. And the beads refer to the nucleosome. Beads. And the term, next one, the term nucleosome was given by, the term nucleosome was given by the scientist Audet. Audet coined the term nucleosome. Now, nucleosomes are described as, they are disc-shaped structure. Now we are going to see, they are disc-shaped structures, disc-shaped with a diameter. They have a diameter of about 11 nanometers, and a height of about, we will take about 6 nanometers. It may be sometimes given in some books as 5.5 also. So 6 or 5.5 nanometers, that is the height. And the nucleosome consists of two parts. There are two parts present in nucleosome. First part is called as a core, or this is also called as a new body, NU. It is called as a new body, or it is called as a core of the nucleosome. So this core of the nucleosome is made up of histone proteins. These histone proteins are 8 in number. There are 8 histone proteins are present, 8 in number, 8 histone proteins. That's why this core is called as octamer, which is made up of 8 histone proteins. So it is called as octamer. And what are those types? There are 4 types, 8 histone proteins, and 4 types are present. They are H2A, H2B, H3 and H4. So these 4 types of histone proteins are present, each with 2 copies, 2 molecules of each. So total it makes 8 histone proteins, and that forms the octamer. And these proteins are basic in nature. They are basic. That is, they consist of amino acids like lysine and arginine. They are made up of amino acid lysine and arginine, which are basic amino acids. So this DNA is associated with histone proteins. DNA is associated with histone proteins. So now how this DNA and histone proteins, they are associated. So I will show you here with the help of a diagram. So we know core of this is made up of, 8 histone proteins, and that means 4 types. Say this is how the 2 molecules of each, I have drawn 4 here, so 2 molecules of each. They are H2A, H3, H2A, H2B, H2A, H2B, they are H3 and H4. This forms a core. Now this core is surrounded by, 2 spirals of DNA. So core we have written here. This is what is the core, and what is the second part? It consists of 2 spirals of DNA. So how these 2 spirals of DNA are present? So they wrap around this, they surround. This is how these spirals they make, completely wrapping or surrounding the core. So I'll make it a double lining. This is how the DNA is completely wrapping around this core, wrapping around. So it is two spirals of DNA which is completely surrounding the nucleosome. And now let us see the details about what are these two spirals of DNA. So these two spirals of DNA which are surrounding this, these are made up of 146 base pairs. That means core consists of DNA with a length of 146 base pairs. And the DNA which comes out of the nucleosome, this is the core of the nucleosome. This DNA which is coming out, this is called as a linker DNA or this is also called spacer DNA. It is called as a linker DNA or spacer DNA. Linker DNA or spacer DNA is of length of 54 base pairs. So this makes altogether the total DNA present in a nucleosome is total DNA present in a nucleosome will be 146 plus 54, that is equal to 200 base pairs. That is the total length of DNA present. Within the core it is 146 base pairs. But outside the core, that is linker DNA, additional 54 base pairs are present. So one nucleosome is associated with about 200 base pairs. So suppose you get a question on this, the number of base pairs present in a nucleosome, you have to give 200 base pairs. Now these two linker DNAs, now once the linker DNA is formed, this linker DNA again can enter into the adjacent. This is one bead-like, this complete nucleosome, this is one bead-like. Similarly, another bead-like structure can also be formed. Another bead-like structure. Bead-like with same H2A, H2B, H3, H4. And this nucleosome, linker DNA, which is coming out of one nucleosome, will enter into the adjacent nucleosome, and similarly it is going to form another two spirals. That's how this appears as a bead-like, string of bead-like structure. So it goes on. So this is the linker or spacer DNA. Now to make this structure very strong and intact, certain bonds are formed. Certain bonds and some associations. So now let us see the bonds which are present between histone and DNA to make this structure very intact or very strong. The bonds are formed. So bond present between histone proteins and DNA. Bond present between histone proteins and DNA. It is called as a salt linkage. The bond is called as salt linkage. The salt linkage is formed between the sugar phosphate of DNA. That means it is attached to sugar and phosphate, and arginine of histone protein. Arginine is amino acid present in histone protein. So arginine of histone and sugar phosphate, they form a bond called salt linkage. So that helps in strong bonding between histone proteins and the DNA. Now another type of association also makes it more strong. Another protein is involved. Other than these 4 types of histone proteins, there is another protein involved which is connecting the two linker DNA. This is called as H1 protein. This protein which is associated, this is called as a H1 protein. So this H1 protein will connect the two adjacent strings. Suppose H1 is absent, then easily the string can break. So where is H1 present? H1 connects two linker DNAs. If H1 is absent, then the structure dissociates. If H1 is absent, then the structure will dissociate. That is how the H1 present. Now, where are these histones actually fit into? Already in my session on DNA structure, I told you the histones will fit into the major groove of DNA. Histone fit into major groove of DNA. They fit into the major groove of DNA. So, histones are positively charged, they are basic in nature, DNA is negatively charged. So, the association between these two will be very, very strong. Now, this is the formation of nucleosome is the first level of packaging. It is called as a first level of packaging. So, before we start the second level, I will give you the number of nucleosomes in human cell. Number of nucleosomes in human cell. Number of nucleosomes in human cell is equal to approximately, in one cell we find 3 into 10 to the power of 7. In human cell we find so many nucleosomes are present. Now this is called as a nucleosome model. It is called as a nucleosome model. So we know in nucleosome model it is the basic first level of folding. It is only the first level of folding, packaging. There are many other levels. So second level when we talk about, this is called as a solenoid model. Second level when we talk about of packaging, it is called as a solenoid model of packaging. So, a solenoid is a term related to physics where a wire is coiled around a hollow core. It forms a hollow core, and around that when a wire is coiled, that structure that is called as a solenoid. So, exactly here also a solenoid structure is formed. So, how this solenoid structure is formed? In the end of the session, I will show a diagram from the book so that you will understand what is it. So, in solenoid model, we have just seen in nucleosomes. So, I will give you a rough diagram. Suppose this is a bead-like structures, nucleosomes are present, and around this there are two spirals coming and then again entering into this, again making two spirals and again entering. This bead-like structure will continue and this will give a long chromatin thread. What is this long thread? It is forming a chromatin thread. We know during cell division, this chromatin is highly condensed into chromosomes. So, from chromatin to chromosomes, there are different levels of folding. So, one of the level, we call it as a solenoid model. So, for this particular packaging, histone protein, that is H1 protein, which is present at the linker DNA, connecting the two linker DNA, this H1 protein is very, very important for further packing. It not only holds the complete structure together, but it also is involved in further packing. So, what are required for further packing is further level or further or higher level of packing is due to H1 protein and non-histone chromosomal proteins. Two types of proteins, H1 protein and non-histone chromosomal protein. So H1 protein, we know the position, it connects the two linker DNA. And non-histone chromosomal proteins are also involved, which further gives stability to the structure. There are three types of non-histone chromosomal proteins which are involved. Non-histone, the full form is non-histone chromosomal proteins. These are further involved in higher level of packing and gives the stability to the structure. There are three types of non-histone proteins, non-histone chromosomal proteins. First is called as a scaffolding proteins. Scaffolding, or they are called as structural proteins. Scaffolding or structural proteins, they actually form the structure. Scaffolding or structural proteins. Second type are called functional proteins. Functional proteins, these are all present in the higher level structures. Functional proteins like DNA polymerase, functional protein like DNA polymerases and RNA polymerases. Already you know the functions of these two. DNA polymerase is involved in DNA replication. RNA polymerase is involved in RNA replication. So these are another type of non-histone proteins. The third type of non-histone proteins are called regulatory type of non-histone proteins. These regulatory type of non-histone proteins they are called HMG or high mobility group proteins. These proteins are involved during gene expression process. Regulation of gene expression. These are called regulatory proteins. So these are required for regulation of gene expression. So all these types of non-histone proteins are associated for higher level of packaging. Now in the solenoid, so what is this solenoid packaging? So imagine this bead-like structure further comes very close, it further folds and comes very close. So this, it appears as a thin thread like with beads. You can imagine further when these beads come very close to each other. They come very close to each other, so that the chromatin appears further coiled. It appears very further coiled. And the beads will come further very. So in this each one there may be many beads present. I will give another color. So each one there will be many beads present. Similarly here also there can be many beads. And in this way each folding there can be many such bead-like structures can come together. And one such type of folding in solenoid where about 5 to 6 nucleosomes will come together. In solenoid, about 5 to 6 nucleosomes, they come together. That is in the solenoid model. And in chromatin fiber, in nucleosome model, we have seen the thickness or diameter is about 11 nanometers. 11 or 10 also we can take for approximation. 10 nanometers thickness we find in a nucleosome. When nucleosome is converted to solenoid, further coiling, solenoid, this thickness will increase to 30 nanometers. So 3 times the thickness will increase, and 5 to 6 nucleosomes will come together. So here all of you are familiar with a diagram given in the textbook. So the same diagram I am going to discuss and then explain. Just have a look at this diagram, all of you. So here what we can find, this is the double helix of DNA, which is of 2 nanometers diameter of DNA. This 2 nanometers, when it is forming nucleosome, histones and nucleosome, it is changing to 10 nanometers. And it is further folding. So how this folding occurs? This is solenoid, which is becoming 30 nanometers. Then further folding will change into 300 nanometers. And further it becomes a chromatid of 700 nanometers. And finally it forms a chromosome. Both the chromatids together, it is 1400 nanometers. So this type of increase in thickness is because of further coiling. That is what we describe in cell division as coiling or condensation. Coiling or condensation. And this coiling or this diagram I am sure you will find in any of the materials children you study. Otherwise you can just write these also. I will write a summary of these in the end of this next so that you will know. So this is how the thickening or condensation process will take place. So during this process of condensation, so first before that I will write the sequence how the condensation is taking place. Starting with DNA. If we start with DNA is about 2 nanometers. This 2 nanometers DNA changes to nucleoid. Nucleoid is of about 10 nanometers. This nucleoid is changing further to solenoid, which is of 30 nanometers. 30 nanometer, this is a nucleosome, not nucleoid. You take it as nucleosome. Then solenoid. And then solenoid, next we take it as super solenoid. Super solenoid. This is about 300 nanometers. This super solenoid is changing to chromatid. One chromatid which is of about 700 nanometers. Chromatid is further changing to chromosome. Chromatid is further changing to chromosome with 2 chromatids. Chromosome means you have to take with 2 chromatids. Then it is becoming 1400 nanometers. This is how the packaging process will take place. Now H1 proteins, how they are involved. Here the most important role is played by H1 protein to form this packaging or compact structures, H1 proteins. So what is the role of H1 proteins? The 2 adjacent H1 proteins will interact with each other. Interact means they just form bond. 2 adjacent will come together. And solenoid, I told you, a core like structure. So all these H1 proteins, occupy central core of solenoid. Central core of solenoid, all the H1 protein. For understanding a rough diagram, we can just have, suppose a hollow cylinder. If we just draw, in the center we can give all the H1 proteins which are all connected, and here we can find all the nucleosomes forming a solenoid, all the nucleosomes which are coming together, all the nucleosomes. And in the center we find this is the H1 protein, and it is H1 protein, you know, is connected to the linker DNA. So this is how you have to imagine this structure. All H1s are present, they come form the center, in the hollow center, and complete the outside core, outside. This is formed by all the nucleosomes. That's how a solenoid and a super solenoid will be formed. And finally, the packaging will be completed. This is what is the solenoid model. Now, in the end of the session, I want to discuss, because we have already discussed the solenoid model, here you have to know what is called as the packing ratio. Packing ratio. Packing ratio of chromatin or DNA. Packing ratio of chromatin or DNA. So what is the meaning of this packing ratio? Means how many times it is folding, how many times it is undergoing condensation. So here briefly I will give you how you can understand the packing ratio, how to calculate, I tell you. So calculation of packing ratio. Calculation of a packing ratio. So nucleosome, we will calculate by starting with nucleosome. So a nucleosome, if we consider nucleosome, it has a height of, we can take say about 6 nanometers and a diameter of about 11 nanometers. Diameter of about 11 nanometers. So for packing we will consider only the height. We will consider, let us consider 6 nanometers. And each nucleosome, if we consider, each nucleosome core has 146 base pairs. Each nucleosome core has 146 base pair. This 146 base pair, suppose we convert into nanometers, we can convert in this way 3.4 into 10 by 10 into 146, because 10 base pairs will have a length of 3.4 nanometers. So we are calculating for 146 base pairs. So this will come approximately 50 nanometers. This will come approximately as 50 nanometers. So packing ratio of a nucleosome. So packing ratio of a nucleosome, how do we calculate? This we have to divide it. This number which we got, the length, we have to divide by its thickness, that is 6 nanometers. The height is about 6 nanometers. So when we divide this 50 divided by 6, we are going to get approximately about 8.33. This is called as a packing ratio. We are dividing the length or number of base pairs present, which we have converted into nanometers, from the dividing the height of 6 nanometers, height of the nucleosome. So for one nucleosome we got a packing ratio of about 8.33. Now if we take solenoid, we know solenoid will have about a solenoid will have how many nucleosomes? We have just seen, we have just discussed. Solenoid has about 5 to 6 nucleosomes. A solenoid has about 5 to 6 nucleosomes. So now if we take the higher number, that is 6, For solenoid, we can calculate easily the packing ratio. So packing ratio of solenoid will be equal to, suppose we take 6, we can multiply simply the packing ratio of each nucleosome into 6. So this approximately answer comes as 49.98, or we can take it as a 50. So packing ratio of a solenoid is taken as packing ratio of solenoid is equal to, we write it as 1:50. That means 50 times the folding has taken place. 50 times the folding of DNA has taken place. In this way, sometimes we find, because there is a further coiling, solenoid, it just doesn't stop at solenoid. It goes to super solenoid and further coiling. So in that condition, the packing ratio, or this packing fraction also it is called, the packing ratio sometimes can reach up to, the packing ratio can even reach up to, in a chromosome, packing ratio can be 1:5,000 also. It can reach up to 1:5,000, which is 100 times more than the solenoid. So this packing ratio, if we take, which it can reach for further completely condensed chromosomes, we find in metaphase, completely condensed. So in that case, the packing ratio can reach even up to 1:5,000. That means it is 100 times more than the solenoid. This is how the packing ratio can be calculated. So I think with this we can conclude this video, my dear students. I hope you have followed. So to understand any topic, my sincere request is to watch the video from the beginning till the end. Once again I repeat, this is not an entertainment channel or an entertainment video where we can keep on forwarding and then we can still understand. This is a educational video. You have to understand a particular topic. If you have to understand a topic, you have to definitely read or understand or view the channel, see the channel right from the beginning up to the end. Then you can understand any topic very, very clearly. I hope it is very clear. Solenoid is very important for many exams, many entrance exams and higher exams also. In my videos now, I want to go further little higher so that it should be useful to any other students, degree students or further higher, those are going for higher studies, and also my dear friends, my youngsters who are coming to this profession newly. It will be benefit to them also. I hope I'm reaching them also. So my only intention is to reach the maximum number of the enthusiasts who want to learn the topic, learn the subject. So that's all for now. So those are again watching my video for the first time, I sincerely request please subscribe my channel and increase my viewer range. That is only my main intention here. If I get a broader viewers from all over, that is the happiest moment for me. So thank you very much. Let's conclude here. We will meet again in my next session. Have a good day. Just a very, very small information, my dear viewers. I just wanted to give you information about the types of DNA. Just I have listed down a table so that which easily you can understand. I have listed down here A, B, C, Z and D types of DNA. Their helix type, right-handed or left-handed. Properties, number of base pairs per turn. Each one have listed down number of base pairs per turn. Location, where it is found, where all it is found, and then length of helix in Armstrongs and diameter. This type of table will be useful for your all entrance exams. So I just wanted to just display this table here so that you can watch and then go through it. So thank you once again, my dear students. Have a good day.